I propose that we still give it 2 or 3 more minutes.
Okay, all right.
Given that mail deliveries have failed as yet, and I have yet to receive any bagels. So we should give it under 2 or 3 minutes.
Okay.
I think I should deliver that to you in person.
Everyone for joining our last session of the lab workshop. This is the second lab workshop. In this last session, there are 2 presentations, and the first presentation is by Prof. Daniel Goldman. He is an expert of microvasculature, modeling, especially about things such as how oxygen and related quantities move in tissues under health and disease. So without further ado, I will pass the floor to Prof. Goldman.
I think I need sharing permission.
Which I will do now. Done.
All right, here I go. I see my presentation. Share it. Okay. And now I just need to hit the right button. Okay, here we go. All right, so hello everyone. Sanjay, thank you for setting up this meeting. Thank you for the invitation. And so here I'm going to talk about the microcirculation, about the structure of the microcirculation, blood flow in the microcirculation, and oxygen transport. So a lot of this will be basic information that some of you may know, especially some of the experts like Jeff and Sanjay. But I thought I should just go over the basics for the benefit of some of the attendees here. And so I will talk a little bit about experimental approaches and also about theoretical and modeling approaches. Oops. Okay, so the outline is the basics of microcirculatory structure, hemodynamics, and oxygen transport, then a brief introduction to experimental microcirculation, some examples of experiment-based microcirculatory modeling, and then talk about some work in progress. Okay, so there'll be sort of 4 aspects to this. The longest will probably be the first. And so what's my perspective in all this? So I'm talking about this subject because this is what I do research in. And so my research perspective is that the microcirculation is a critical organ. So I think of it as sort of like an organ, even though it's different in every tissue and organ. And it's extremely heterogeneous in structure and hemodynamics. Okay, so that's something that's really critical about the microcirculation. One implication of this heterogeneity is that you need a sophisticated regulation system for blood flow and oxygen delivery. We'll see that things are really complex in the microcirculation. And some people think of it as a black box. A lot of times, physiology and medicine consider it a black box. But the details of the microcirculation, I think, are important. And that's something we need to deal with. So understanding the effects of this heterogeneity on tissue under normal and pathological conditions, as I see it, requires a combination of experiments and modeling. Okay. And so that's what I'm going to talk about today. And that's my approach in my research. And so just to take a broad introduction, what is the microcirculation? So on the left here, this is a diagram from a physiology textbook, this Mormon and Heller. And they list the different vessel types in the circulation, right? From arteries, arterioles, capillaries, venules, and veins, okay? The microcirculation is now these smallest vessels in the middle here, from arterioles, which are really small arteries, to the smallest vessels, the capillaries, to the venules, which are the smallest veins, okay? And so the microcirculation is basically vessels less than about 300 microns in diameter. The smallest ones, the capillaries, are 5 to 10 microns. And so just to briefly talk about the function, capillaries are involved in blood tissue exchange. Okay, so of oxygen mainly, but of other things, including water and other solutes like glucose. Arterioles are a little bit bigger. They can change their diameters actively. They have smooth muscle, and they're involved in regulating resistance of the vasculature and in local flow distribution. Okay, so when we talk about regulation of the microcirculation, we're mostly talking about arterioles. Okay, and then venules are the vessels you don't hear much about. They're small veins. They have to do with immune cells getting in and out of the tissues, and they also have to do with regulating blood volume and the capacitance of the vasculature. Okay, so that's just a general introduction to the microcirculation. Okay, and so one thing to keep in mind is that blood flow in the microcirculation is very different from in the macro, okay, the macrocirculation. So you can't really just think everything's smaller. There's really different things going on in the microcirculation. And for one thing, viscosity is really important in the microcirculation. So in the larger vessels, larger arteries, there's a lot of inertia, there's a lot of dynamic things going on, but microvessels are dominated by viscosity, okay? Even though there's complexity, viscosity is very important. And there's something called the particulate nature of blood. So there's a lot of different objects in the blood. There's white cells, there's platelets, but the flow effects are mostly determined by having liquid plasma and then the red blood cells, okay? And so when we say discrete cell effects, we mean red blood cell effects in the microcirculation. And these are seen because you're getting down to vessel sizes that are around a red cell size. So on the order of 10 microns or tens of microns, maybe a couple hundred microns. And so you're getting discrete cell effects that you don't get in large vessels. Okay. Oops, is that it? That's all I wanted to say there. Okay, and so another important thing about the microcirculation is that we talk about networks, okay? And so the microcirculation is arranged into networks or functional groupings of vessels, so of arterioles, capillaries, and venules. Over here, you see kind of a very classic schematic that you might see in a physiology book of an arteriole supplying a bunch of capillaries, and then the capillaries are being drained by this venule. Okay, so this basic unit is seen a lot and talked about a lot, but there are other types of networks. Okay, so this is a capillary module or capillary network. There are other types of networks that I'll show. And so understanding capillary networks and larger microvascular networks is believed to be fundamental to understanding microvascular function or microcirculatory function. Okay, and so you really can't just look at individual vessels and say, I know what every capillary is doing, because they only really function as a group. And the same with arterioles and venules. You can't just say, I know what one arteriole is doing. They usually are in networks that interact in different ways through blood flow, through signaling, and other things through solutes. So there's lots of ways that networks function as a bunch of vessels, and they can't just be looked at in isolation in terms of each vessel. Okay. So just to give you an idea of the complexity of the microcirculation, and I'm really focused on muscle because I've mostly worked on skeletal muscle. Here's just some examples of experiments that have been done. So this now is from a mouse leg muscle, and you can see that depending on the scale you look at, you can either see a bunch of arterioles, you can see very dense capillaries here, right? All these little lines are capillaries. And then if you look over here, what's shown is from the cat sartorius muscle, okay? And there's different levels here. And so this basically is larger arterioles or maybe very small arteries. It's got a structure something like this. And then branching off this, which you could call it an arcade, is these branching networks of arterioles that I'll talk about later, okay? And then off of these arterioles, you get capillary networks, okay? And so this little star means that there's something like this sitting in there. And then these two stars mean there's something like this sitting there, okay? So there's different levels of organization that you could consider when you're trying to study microvascular networks. And so I'll start with arteriolar networks, which in a certain length range of diameters or length scales are fractal and branch, okay? And so the diameters and lengths decrease exponentially as the branching increases, okay? And so, for example, here's some data from a paper by Al Tarhouni and coworkers with myself and another researcher. And measuring diameters, you get this exponential decay, okay? And that's characteristic of these branching networks that in the end, if you map them out and you put them into some software like ImageJ, you can get a fractal dimension, okay? And depending on which animal you're looking at, which muscle, you may get slightly different fractal dimensions, but it just shows that they have this tendency to be fractal the way they branch, okay? And so arteriolar networks definitely are fractal to some extent. And now yesterday it came up. Now, I didn't see Gabrielle's talk, but I did watch the video on YouTube. So that came in to be important. And I heard I think Dr. Frisbee asked a question about ordering. And so when you're ordering these branching networks of arterioles, there's basically two ways to do this. There's a Strahler order where you find the terminal arterioles and make them ones. And as they go up, you get higher orders, okay? Or there's this centrifugal order where you start at a large vessel, and as it branches down, you get to higher orders, okay? So there's two different ways to order things. Some say the Strahler order is kind of the most logical, but the centrifugal order partially comes from the fact that if you start at large arterioles and look down the network, if you can't get to capillaries, you don't really know where the ones are. And so you just have to decide how far you want to go and some way of labeling the different orders going down the tree. Okay. And so now we can talk about capillary networks. So capillary networks do have a 3D structure. So this is from the EDL muscle. And capillaries are space filling. So they don't tend to have a fractal kind of geometry. So they're networks with branchings, but there's only a small change in vessel number, diameter, and length as you go from one end to the other. Okay, so they don't really have a fractal dimension. They do have a very small scale. They have complex geometries. And so this is one reason why we might try to simplify capillary networks into some continuum description. So that's something I'll talk about later. Now hemodynamics, we can talk about hemodynamics and microvascular networks. And even though I said the microcirculation is much different, there's a lot of things that are similar to larger vessels. So for example, flow is determined by pressures and resistances, okay? So if you have your network structure and you have your pressure boundary conditions and the resistances of all the elements, you can find the flow distribution, okay? And now one thing that's different about the microcirculation is that pulsatility from the heart is mostly damped out, right? So in arteries, larger arteries, you have these oscillations in pressure that come from the heart. But in the microcirculation, for the most part, this pulsatility is gone, okay? Now you do have oscillations, but these mostly occur because of blood rheology. So it turns out that just the way red cells distribute, you can get some oscillatory behavior, and also from regulation, okay? So when arterioles try to regulate to say get a certain flow in a certain place, then that can cause oscillations, okay? And now resistance is a little different in the microcirculation because although it does depend on length and diameter, following Poiseuille's law, so resistance goes like viscosity, length over diameter to the fourth. There's also network structure that can affect effective resistance, and there's also blood viscosity that can be variable. Okay, so this is something I'll talk about. So essentially in the microcirculation, there are three effects that you see in small vessels you don't see in large vessels, and these are what produce heterogeneity and require really sophisticated regulation to make things work, okay? So now let me maybe pause for a second. So does anyone have questions? I'm willing to take questions either orally or in the chat. I don't think I have my chat open. Does anyone want to stop me and ask a question? No? Okay, all right, well, feel free to do that as I continue here. Okay, so the first thing I'm going to talk about is called the Faraeus effect. So this has to do with the fact that hematocrit actually decreases in the microcirculation. So tube hematocrit is volume average. So you just take a snapshot of all the blood in a vessel, some of its plasma, some of its red cells. The red cell fraction is the tube hematocrit, okay? But if you measure tube hematocrit in microvessels, so this is large venules here, large arterioles going down to the capillaries in the middle, as diameter decreases, effectively the hematocrit decreases, okay? So this is something strange that people didn't understand maybe 100 years ago, but it's this effect where the tube hematocrit decreases on average as the vessels get smaller, okay? And this is seen in glass tubes. I think it was first seen in glass tubes of just like small diameter, like 50 microns. It's seen in isolated microvessels. And it's also seen in intact tissue. So this result is from intact tissue. And so it doesn't depend on, the biology really of the walls. It just depends on the red cells and the fact that the vessels are very small. Okay. And so what causes this? So my other perspective on all this is biophysics. Okay, so I'm trying to understand the biology. I'll show a bunch of equations and things, but also I'm trying to connect physical processes to what we see biologically. And so if we see a decrease in hematocrit here, we'd like to know why is that happening. And it's not happening because you're losing red cells. It's happening because there's this plasma layer near the wall. And as the vessels get smaller, that plasma layer takes up a bigger and bigger fraction of the vessel cross-section. And so essentially that leads to lower effective hematocrits. Okay. And what happens is the red cells at the center flow really fast. And so even though they have a low volume density, right, they still have a high flow rate. And so the red cell velocity comes out to be the blood velocity times this ratio of hematocrits. Okay, so tube hematocrit is your normal hematocrit. That's what's going down here. Discharge hematocrit is something that's flow weighted, and it tends to be bigger than HT in situations like this. And so since HD over HT is greater than 1, you get that your red cell velocity is higher than your blood velocity. Okay, and so you could flip this around, and this would explain, why HD is greater than HT, because your red cell velocity is higher than your blood velocity. Okay? And so this is from the biophysics, right? There's plasma near the wall. It has low velocity because there's no slip at the wall. There's no velocity at the wall. But in the center are red cells, and they're moving very fast. And so that's why you have this effect. Okay? So that's the Faraeus effect. Hematocrit's going down. This Faraeus-Lindquist effect has to do with the viscosity is actually going down also. So if you fix hematocrit, right? Hematocrit affects viscosity, but if you fix it and then you go into smaller and smaller tubes, you're going to get lower and lower viscosity. Okay? And this is starting maybe around 1 millimeter diameter. So you don't see it in large vessels, right? These curves are flat here as diameter gets big, but as diameter gets small, you see a decrease in viscosity. And so again, we see the same effect in glass tubes, in microvessels that are isolated, and in networks. Okay? And so it's persistent effect that's really there. And it's related to this plasma layer near the wall and this Faraeus effect decreasing tube hematocrit. Okay? So hematocrit affects viscosity. If you decrease your effective hematocrit, then you decrease your effective viscosity. Okay? And so that's the explanation. And one thing to keep in mind is over here, you see viscosity going up. So that's called the reverse Faraeus-Lindquist effect. So as vessels get really small, like small capillaries, red cells have to squeeze through, the plasma layer gets squished, gets smaller. And so you get back to the situation where you have systemic hematocrit and systemic viscosity. Okay, so reverse Faraeus-Lindquist and reverse Faraeus effect. Okay, and so the last effect I'll talk about, probably that you guys have thought about or heard about, is this phase separation or plasma skimming. Okay, and so what this is that when you have flow into a diverging bifurcation, then you don't get the same distribution of red cells to both branches. Okay, and so the one branch might get a higher concentration of red cells compared to the other, and that will change the hematocrit in the system. Okay, and so at the diverging bifurcations, red cells prefer the higher flow branch, and so that changes hematocrits. Okay. Now, again, why should hematocrit care, how much flow goes to each branch, right? So if you have a large artery, you have a division of flow, the diverging bifurcation, those two branches will still have the same hematocrit. Okay? It doesn't matter if one's really small, one's really big, hematocrit won't change in large vessels. But in small vessels, you have this plasma layer near the wall, you actually have higher concentration of red cells in the center than as you go out. And so if you take a small amount of red cells just near the wall, that will be low hematocrit, okay? And if most of the cells going this way are from the center of the vessel, they'll have higher hematocrit. And so that's why you get this effect, okay? Because of the radial gradient of red cell concentration. And there's different ways to look at it. Here you can see the two vessels. Depending on the diameters, so one now means the daughter vessel has the same hematocrit as the parent. So you can see here, this is a small vessel here, beta, a big vessel, alpha. And so for beta to have the same hematocrit as the parent, it needs a small flow, maybe 35%. And for alpha to have the same relative hematocrit as the parent, you need maybe a 60 to 65% flow to that vessel, okay? And so basically where that set point is depends on the size of the two vessels. But in the end, what you get is that the fractional red flow to the two vessels is usually not the same as the fractional blood flow, okay? So if we're sitting, let's say even at 50%, right? So this one vessel is getting 50% of blood flow, but it's, right? Yes, but it's getting less than 50% of red cell flow, whereas this one is getting more than 50% of red cell flow. So this effect isn't that great, but it leads to heterogeneity of hematocrit in the different microvessels. Okay, so it's something to keep in mind. And as you go through a branching network, if this keeps happening, then you end up with a big spread of hematocrits as you go downstream. Eventually, right, the smallest arterioles feed into capillary networks or capillary beds. So if you have heterogeneity in the hematocrit in the arterioles, then each capillary bed will have a different hematocrit coming in. Okay? And so then the question is, how do you regulate oxygen delivery to capillary beds if the hematocrits aren't even the same, right? So you can't just regulate blood flow. You actually have to take into account how many red cells are they getting, maybe even how much oxygen do these red cells have. And so that's why you need a sophisticated regulation system for microvascular blood flow. Okay. And again, this effect depends on the size of vessels and the hematocrits of the vessels. Okay. All right, so that was my intro to microvascular physiology or microvascular biophysics. Any questions again? Anyone wants to ask? Okay. Oh.
Can I interrupt, please?
Sure, Dr. Karche.
Sorry, I'm a major waste of time, but I will do that anyway. On the previous slide, you described this bifurcation anisotropy here.
Yes.
And so in view of when such bifurcation anisotropy must exist, do you expect for small blood vessels to regenerate themselves periodically?
Regenerate?
Yes.
You mean form anew or change their
Yes,
size?
yes. Forming new vessels.
But I think that happens from the capillary end. So in other words, a 9-micron arteriole probably isn't going to just go away, but it's downstream capillaries. They may, more of them may form or less of them may form. Sorry, Dr. Frisbee?
Oh, I don't know if you had more to say about Sanjay's question.
Sorry, Sanjay, that's the thing. I think this mostly happens at the capillary level, unless there's damage or something.
Sure.
That's how I see it.
Thanks.
Sure.
So Dan, you made a couple statements. I wonder if you could maybe clarify, because I just think it's kind of interesting. You said that hematocrit may be a regulated variable. So tube hematocrit, not discharge. I mean, discharge is just the same as systemic generally.
Yeah.
But is there evidence that hematocrit is a regulated variable?
In capillary beds, yes.
Can you briefly describe it? Because I hear different sides of this argument. So I'm curious, that what is it that makes hematocrit a regulated variable as opposed to something like perimitochondrial oxygen tension being a regulated variable?
Well, a regulated variable, I guess I mean a regulatable variable. I mean, in other words, if you have a capillary network,
yeah,
or a few capillary networks, and they need more oxygen, then they need more blood flow, which means they need more red cells. And so they can get more red cells, right, by just higher velocity with the same hematocrit. But they can also get more red cells by maybe a little change in velocity, but also a change in hematocrit.
But does that mean that hematocrit's not regulated? It is. You can get there in multiple different ways.
Right,
but
so I don't think it's fixed. So I wouldn't say we fix the hematocrit going to different capillary beds, no. But if we need to increase it or decrease it, we can do that dynamically.
yeah, I guess I conceptualize this a little differently. So that's, and I think we're probably saying similar things, just using different jargon. But yeah, I wonder if, I actually don't think hematocrit is a regulated variable at all. Like, I think it is just what the outcome happens to be. Like, it's the regulated variables are much more metabolic, in my opinion. And then the hemodynamic effects become just ways to get to that metabolic outcome. But as you said a second ago, you can have, you can change oxygen delivery through a variety of mechanisms. It all does come down to hematocrit having to go up. But whether that's the regulated variable, I guess I want to know, what would the sensor be if that's going to be the regulated variable?
Well, I guess in a sense, what you said in the beginning was that it's almost like the system doesn't care about hematocrit. I mean,
it doesn't regulate it. Yeah.
Doesn't care about it directly, right?
Yeah.
And so, for example, this picture I'm showing here, if this was, say, a terminal arteriole with its final branch,
Yep.
and there was a capillary bed here,
Yep.
and if the capillary bed was all happy with oxygen delivery, right, everything would stay the same, and it would be getting some sort of random hematocrit, but
Sure.
enough red cell flow and oxygen. If it needed more red cell flow and oxygen, then something has to change here. So the flow here could change, and you could get extra here with the same distribution of red cells. But if this vessel dilates a little bit, it's going to actually pull in, besides a little more flow, more red cells with higher hematocrit.
Absolutely, yes.
And so, I mean, these things happen. I don't know how, well controlled they are.
Yeah, so I guess my question is, the term regulated, is that the outcome that's being regulated? And that's where my question sits. But I couldn't think of anybody who's demonstrated that. That's why I was wondering why you said that. So
right. I mean, I guess it can be part of the regulation system.
Yeah.
But I guess when you say regulated, that it's like mean arterial pressure, like it should be fixed at something, and the system needs to keep it there, right?
Yeah. No, these are just, we're not going to get to an answer to this question in the next 3 minutes, but we'll probably both be dead before they come up with one. But that's enough for now.
Okay, thanks, Jeff. I appreciate all questions. Okay, so now I'll create more controversy by trying to talk about experimental work, which I don't do. So this is just my perspective. Yep, go ahead,
Hello,
Daniel.
sorry. Could you think you go back to the last slide? I'm just curious. I'm second year, so maybe I'm not as knowledgeable as all of you, but do you think the body could control the amount of hematocrit that gets to specific areas by controlling the vessel diameter?
Right. So as I said, if this thing dilates, it will probably pull higher hematocrit down in that direction. So locally, I mean, the thing is, it's not like your brain is saying, my fingertip needs a higher hematocrit, and it sends it there. But things going on locally may serve to change that hematocrit in your fingertip.
But unconsciously, it could.
Right. So, I mean, part of the microcirculation, I think what most people believe is that, I mean, there are local regulation mechanisms that are doing something in some way, without involving the systemic nervous system or whatever. And so that's, changing hematocrit is something that I think can be done locally.
Thank you.
Gabrielle has a question in the chat.
Gabrielle, let me see. I can read the chat. Ordering. Okay.
She wonders if you could go over the ordering that was mentioned.
Right. Okay.
I guess she wants to do ordering of her vasculature network.
Well, this is something. The details of this definitely she can find somewhere. I'm not the expert on this, but just the basic idea is that if you have a branching arteriolar tree here, right, from large to small, if you can find where it feeds capillaries, you can call those one. And then if those join up, they form higher orders. And so that's the strahler. Okay, so the ones feed capillary beds, they're terminal arterioles. And that's kind of the most standard way. The centrifugal, you just decide that the largest arteriole in a small muscle is 1. And then as it branches, you increase the order. And eventually you get to capillaries, but you may not know exactly where you get to capillaries, right? So here, if these are the same networks, okay, 6 is feeding capillaries, but here it's 5 that's feeding capillaries. So it's a little more ambiguous when you do the centrifugal ordering. So is that good, Gabrielle? Okay, yeah, and you can find papers where they talk about the rules for doing this, which I think is probably the most important part, practically speaking. Okay, let me see if I can get back. Okay, all right, so here I'm diving into experimental. And since this was in my title, I thought I should talk about it. Other people will probably tell me I don't know that much about this, but anyway, we need to measure the microcirculation, right? Just like any part of biology or physiology, we need to do measurements. And people have developed many techniques, I would say, over the past 100 years. And we want to measure vessels, length and diameter, network structure, blood flow, oxygen transport. These are things we'd like to measure in the microcirculation. And so people have developed all kinds of different methods. To initially get geometry, they did this casting, right? So you can, in an animal muscle or something, you can put some plastic in there, it hardens, and you can use that to figure out what the structure is, okay? You can somehow measure that cast. And nowadays you can do staining and even, sectioning and find out exactly like where the endothelial cells are and all kinds of things like that, the mitochondria. And then, so dynamically though, one way to do things is to look at isolated vessels like arterioles, and you can isolate them, you can perfuse them, stimulate them, and see how they respond. And that's a way to understand part of the regulation system. You can also look at the electrophysiology of vessels. This is something I know Dr. Cartier is interested in, and find out what they're doing in terms of sending signals along the vessels. In terms of oxygen, you can do things like electrodes. I think some of you have probably taken 3507. We talk about putting a PO2 electrode into a tissue that can measure O2. There are dyes, and a fairly recent method for this is called phosphorescence quenching. So you put a dye in there, but how fast, when you excite it with light, how fast it decays depends on the oxygen concentration. And so that's a good way to measure oxygenation in vessels and tissue. And then ultrasound, which I guess anyone in biophysics learns about ultrasound, that's a way to measure velocity in red cells in the microcirculation. So there's lots of ways to do this. My favorite way, since it's the way that people I've worked with have used, is intravital microscopy, okay? And so this is nice because you have a live tissue like a muscle, you can watch it dynamically, you can watch the red cells flow, and if you work hard enough, you can get some geometry out. And so this is one way to measure what's going on in the microcirculation. The drawback of this is limited depth, right? Because you just have light going in and out or light coming through. And so the depth is 100 microns or so. Nowadays, they have dual photon microscopy. So they're really high-tech methods to try to get deeper in the tissue and doing the same kind of thing. But IVVM or just IVM is very, very nice when it works. And it does work well for thin muscles. So muscles in small animals like the rat, the mouse, if you can transilluminate them and you can see the vessels and the red cells going through, you can get a lot of information from that. And I think probably what Gabrielle showed yesterday was IVVM data. And so there's a lot of people who do this. And so now let me just, hold on. So I'm going to talk about using IVVM on to get capillary information and also to get arteriolar information. So for capillaries, so this is a method developed by Chris Ellis, who's at Western or who's emeritus now. But basically, you can look at vessels, a capillary oriented this way, and you can watch the red cells go through. And there are ways to pull out the velocity of those red cells, the lineal density. So the slopes here are basically velocities. If you go this way, you get lineal density. If you go that way, you can get supply rate, okay? And basically, velocity times lineal density gives you supply rate. So you can get information about red cells in capillaries. And these are actual images of vessels. So this was a cartoon. And basically this is one vessel over time. And as the red cells go through, they make these little streaks. And so the slopes will tell you velocity, and you can see that you can get densities and things like that. So this gives really good hemodynamic information about individual capillaries. And if you have a field of view, if you look at enough capillaries, you get an overall picture of red cell velocities and supply rates and things. Okay? So that's individual capillaries. And then Chris Ellis and I had a student, Graham Fraser, a few years ago, who put together the system that measures individual capillary data. And we can put together small networks now, where we have capillary geometry and also flow data. So this is red cell velocity in this network, okay, at the surface of the EDL muscle. And so this is fairly good information for trying to understand things like oxygen transport. Okay? So that's IVVM for capillaries. And then you can do a similar thing in arterioles. Now, in capillaries, you have single cell flow, so you can watch those red cells go through. In arterioles, you could have in the cross section, tens of red cells, and you can't really watch individual ones go through. But if you label a small fraction of red cells, I think it's like 2% or something, then you can look through the microscope and you can watch streaks go. Okay, so if you excite the red cells at the right wavelength, you can observe at another wavelength, and you can see the light from these streaks, and you can actually take snapshots, especially with good cameras that we have now, digital cameras. You can measure the length of these streaks, and that gives you the velocity of the red cells. Okay, and then what you do is you can measure a bunch of different streaks across the vessel. You get a velocity profile, okay, and then that gives you the flow rate. You can integrate that profile, get a flow rate for blood in that vessel. And then you can also use the density of these streaks to get hematocrit. Okay? And so that's a way to measure flow in intact tissues like the gluteus maximus muscle at the arteriolar level. And using similar techniques, you can go through, and now this is really painstaking, but this is about 500 fields of view that were put together by Mohammed Al-Tarhuni, and he reconstructed actually the arteriolar tree is the smaller lines, and the fatter lines is the venular tree in this muscle. Okay, so the length scale is 1,000 microns. I believe this whole thing is about 1.5 by 2 centimeters. With a vessel here, an arteriole of about 150 microns, and then the smallest arterioles we find are, I think, around 20 microns. So you get over a fairly large range, you get this branching structure of the arterioles and the venules. And so before I go on, let me ask again about questions. Okay, does anyone have questions about these two slides I just showed with measuring flow in arterioles and capillaries? No?
Dan,
Yep.
Sorry, Jeff, please go ahead.
yeah, Dan, quick question. You're talking about all these different techniques. One of the classic techniques that's been used a lot, which maybe it's limited for what you're trying to do, is tracer washout kinetics. Like, are there still things that we can learn from washout kinetics, and like looking at a spike input and then the venous effluent in terms of flow distribution? Or is that really not going to be that helpful for the kinds of things that you're talking about here?
Yeah, I think probably it has maybe limited use in developing what we want to develop. It could be still good to sort of validate that we have the right structure, right? Because we can measure tons of individual vessels, but if what we get out from the whole flow doesn't agree with the tracer kinetics, then we've messed something up.
So it'd be better just so I guess everyone knows, tracer washout kinetics is when you put a spike input into the arterial side of something that's either radial labeled or fluorescently tagged, and then you measure on the venous side to see how it elutes from the network. And you can do a bunch of deconvolution and really complicated math to determine what is the flow distribution profiles and things like that. But maybe, so I guess my question is that better used as a check of your modeling methods and approaches, and rather than using it as at the front end?
That's what I'm thinking, in that we definitely need this, detailed data of where the vessels are, velocities in individual vessels, to make sure we're getting everything right from the beginning. But as I'm going to show in a little bit, we could screw up the network, and we may have some of the vessels right, but we may have the overall picture wrong. And so I think having the tracer washout kinetics might be helpful for that.
Thank you.
So then my question follows on from what Jeff has just said. Would you comment on if tracer washout kinetics, as used, say, for example, by people in robots, is the best clinical way to measure microvasculature properties? For example, is the mean transit time reported in Tingley's CT scans representative of microvascular health status?
It might be, but I don't know. I think that it's hard to connect those kinds of results to what's really going on at the small scale, of small arterioles and capillaries.
So on either side's measurement or in the modeling side, we need a few more steps before there is any conclusive connection between the clinical observation and the microvasculature status. Is that what you're saying?
I think so, especially given the way they do some of those things.
Thanks, Dan.
Yeah. Thanks. Okay. So if there are no more questions on experimental methods, I'm going to talk a bit about modeling. And so I'm going to just talk about basically two things to start: modeling hemodynamics and modeling oxygen transport. Okay. And so both with the idea that we're looking at microvascular networks. Now, especially in the last 5 to 10 years, people have actually started to do really detailed full 3D time-dependent simulations. You have your Stokes equations, right, of plasma flow in vessels, deformable red cells, and they can basically, mimic what's actually going on in your blood vessels. They have plasma, red cells, it's all there, but it takes a lot of computational work. And there's still assumptions in there that have to be tested. A simpler approach for now, similar to, I guess, what some of our students talked about earlier, in terms of using a 1D model versus a 3D model. So you can do the same thing in microvascular networks, and you can kind of use a nonlinear circuit model where you just have a 1D description of flow in vessels, okay? And then you just apply basic properties like mass conservation, Poiseuille's law for resistance, this viscosity changes, this plasma skimming effect. So you apply all these things, and you can get a model of flow in a network. Right? So basically, flow conservation at any bifurcation. So whatever flows in has to flow out. That's for total volume. And then this is red cell volume. Whatever red cells flow into a bifurcation have to flow out. This is Poiseuille's resistance. And then this is this viscosity law. So the Freitas-Lindquist, where, so now eta is viscosity here. So viscosity depends on vessel diameter and the hematocrit there. Okay, so variable viscosity. And then in addition to this a red cell volume conservation, you need, this what we call the bifurcation law. So this tells you essentially in a diverging bifurcation how red cells are distributed according to the diameters. So the 3 diameters, parent, daughter 1, daughter 2, and then the ratio of one daughter flow to the parent flow. Okay, so if you know all the information and you know the parent hematocrit, you can get out one of the daughter hematocrits, and then you can use this equation to get the other daughter hematocrit. Okay, so you essentially get two sets of equations here, and they're actually linear, but by solving back and forth here, you can eventually converge to a set of blood flows and a set of hematocrits that fully describe what's going on in your network, okay, and at steady state. So this is a steady state model. And so there's nonlinearity here, right, in the viscosity law, in this bifurcation law, but by doing two linear problems, you eventually resolve all nonlinear behavior and get a consistent flow throughout the network. So that's how we model hemodynamics. This model was eventually put together by Fries and Seacombin and coworkers in the 90s. There have been a few updates, but it's mostly the same as it was. So that's the flow model that I mostly use. We can make it time dependent if we want. I'm not going to talk about that really here. And so now what I want to talk about was an example of using this flow model. Okay. And so the example is to sort of figure out the resistance of capillaries and very small arteries, or sorry, very small arterioles that we don't generally measure. Okay. So if you look at this picture here, oops, this picture now, it has these arterioles and venules that we measured by putting together all the fields of view. And we made some attempt to connect terminal arterioles with terminal venules. And we'd like to know, what the flow is in this whole system. But to be able to solve for the flow in the whole system, we need to put in a lot of missing information. Okay, basically, there are a lot of small vessels that we don't capture in our measurements. And then there's also capillary beds downstream of the small vessels that we don't capture. And so trying to somehow put together a reasonable model of this by putting back in the resistances for the arterioles and the capillaries. So we just go to the next step here. Well, so first of all, there are missing things in this network. And so we can calculate the flow. Okay, I had this. So we can calculate the flow, for example, this is in the arterioles. And so we can use our model, and you can see there are lots of little segments here, but we can use our model to calculate this flow, and then we can look at how it agrees with measurements that have been done of flow in this type of network, and how different components like the venules and the capillaries, how that affect the flow. And so this is from Zara Farid's master's thesis from years ago. And she basically took just the arterioles plus capillaries and smaller arterioles that are missing, and then also added in the venules. So you can look at flow in sort of three types of networks: just arterioles with capillaries and smaller vessels, and then adding in the venules. And so, not surprisingly, the venules don't add much to resistance. So this is resistance. And the venules also don't change the flow distribution very much. Okay. And so from this, and from comparison to measured flows, we developed a model for adding in small arterioles and adding in capillaries to this network. And so, as part of that, the flow model was key, because that connects what we measured, the assumptions we made about smaller vessels, and then it gave us the flows in the end. And so then we developed this model, and then we took a small arteriolar network here where we actually measured the flows at these 5 bifurcations. Okay, so for each bifurcation, we got 3 flow measurements. And so then we went through, we took this arteriolar network, we treated it as we would any other one that we measured. We put in small arterioles that we had measured downstream, we put in capillary resistances, and then we calculated the flow and we compared it to measurements. Okay, so this is measured flows in, I think it's 15 vessels, and this is the predicted flows from the model. And we got pretty good agreement, which we took to mostly validate our approach. Okay? And so this approach basically says that we know how to add in missing small arterioles, and we know how to add in capillary beds and their resistances. And in the end, we can calculate flow in an arteriolar network, just given its geometry that we can measure. So maybe I'll take any questions about this. I'm sure someone has some.
Then
Yeah.
on the diagram on the left-hand side, a rather beautiful diagram. Would you comment on the average distance between the red and blue lines?
You mean that it's very small?
What is the average distance between the arterioles and the venules, please?
Okay, so as they're bifurcating here, these distances, you can see they follow each other for a while. Distances are very small. I think now the vessels are about 100 microns. So I would say the distance is maybe a few hundred microns apart.
And so since the red vessels carry oxygen blood and the blue vessels may be carrying that much less oxygenated blood.
Yeah.
Are these two vessels exchanging anything among them other than at the capillary beds?
I think they are. I mean, maybe not a ton because they do have thick walls.
Mhm.
Their walls could be, 20 microns thick.
Mhm.
And then if they're 100, 200 microns apart, then
Yeah.
it could be that they don't exchange a lot up here. Probably right before they start to separate out, when
Mhm.
they get down to 30 or 40 microns in diameter, that's probably where they're exchanging the most oxygen.
Thanks.
That's a really good question, Sanjay. And I think Dan's right. Like as you get up, I'm using my own mouse, but that's kind of silly. Yeah, so I'm assuming at the very top, that's a 1A.
Yes, yeah.
I think that the issue is, Dan's absolutely right. They're not exchanging very much at that level. But as you get down into the 3s and 4s, you will start to get some of that oxygen exchange when they cross, especially when they cross. But the other challenge, there's a pretty good literature on this, although it's been probably 15 years since it's been added to. The venular side is releasing arachidonic acid metabolites, predominantly prostacyclin, or as you get into disease states, you can get into thromboxane, which Robert Hester down at Mississippi has shown really starts to impact arteriolar tone in the paired arteriole that goes with it. So what's happening in the venules is leaking across and impacting arteriolar tone as a contribution to the whole oxygen matching, yada kind of thing. But it really brings up a pretty interesting possibility that you mentioned. Sort of a venular control over local arteriolar function. Robert's moved on to creating the virtual human now, but we should definitely have him as an invited speaker for this kind of a thing because, his work on this, like back in around 2000, early 2000s, was really pretty good. So.
I think it's definitely, even though in this slide, I've said the venules aren't doing much in terms of resistance, I think we still believe that there's basically an interaction between arterioles and venules that is a big part of the regulation system.
Yeah, well, we published that one paper with you a couple of years ago. Yeah, the venules aren't doing much at all. They're just, they're there.
Yeah.
But Hester's thing, so hemodynamically, they're not doing a lot. But in terms of control, that's another story.
And I believe Keith has a question.
Okay, Keith?
Hi, Dr. Goldman. Just a really quick question. I never thought about it until I looked at this wonderful diagram on the left-hand side. And I was just curious, while there's probably not oxygen going from the arterioles to venules through diffusion, a lot of times, in that space, do shunts between the arterial and venal side happen often or not that often, like AV anastomosis or shunts or those kind of things?
So up here, these vessels are pretty large for microvessels, and I don't think there's shunting of flow. There could actually be, very small vessels coming off here that we're not seeing that supply some small capillaries that are in there. So that's probably what's going on. I don't think there's too much shunting at these large vessels.
Okay.
Yeah.
Okay. Thank you very much.
Okay, so that's my flow example, and now let me see if I can get to talking a little bit about transport. Okay, so now I'm talking about oxygen transport and involving tissue and microvascular networks. And so again, to do transport, you could consider something very complicated. You could consider discrete red cells and vessels, discrete elements in the tissue like cells and mitochondria and membranes and things. And I think this brings up that basically we're always approximating. And even if we don't think we're approximating because we really don't know everything that's going on in these, biologically. And so this would be a very complex model. But again, we've taken a simpler approach where we use our two-phase continuum flow model that I just described, and we use vessels, these vessels embedded in tissue that we assume is continuous. And so it's basically a continuous medium for diffusion, but it also consumes oxygen. And then we do put in, so people have found that there is a discrete effect in oxygen transport between vessels and tissue. And so to take care of that, we put in this mass transfer coefficient that I'll describe. And so these are, you'll see these be convection equations and diffusion equations. And in the blood, it's mostly convection. In the tissue, it's diffusion and consumption. And then we sort of have two domains, two continuum domains that we connect by the boundary conditions. And so here are some of these nice equations that Sanjay gave a preview of. And, you know, they look complicated, and they could be simplified a lot and without missing much. So basically, you know, this is the blood saturation of oxygen. This is hematocrit. This is the binding capacity of red cell hemoglobin. And so this is the dissolved oxygen in the blood, which is really small. So you could just drop all these alpha terms because there's not much oxygen carried in the blood in dissolved form. This is the blood velocity. So velocity times a gradient along the vessel. So that's the main convective transport term. And then this is the wall flux J. So that's as a vessel flows along, whether it's an arteriolar or a capillary, it loses oxygen through the wall. And so that term takes care of that. And then this is time dependent, but for most problems, you can just get a steady state solution. So you set this to 0. Now this is for tissue. So this is the PO2 in the tissue. So that's how we describe O2 in the tissue as a partial pressure of oxygen. In this case, if there's myoglobin in the tissue, you do need to consider that. And most muscle has myoglobin. So you do sort of need this complexity, at least for time-dependent terms. And if you're going to have low oxygen anywhere, you might need myoglobin. On the right-hand side, we have now, this is a consumption term. So O2 consumption can depend on the PO2. You could also use a constant value. This is now diffusion, which is usually 3D initially. And then, so this is diffusion of free oxygen. And then this is now a diffusion term for oxygen bound to myoglobin. So when oxygen binds to myoglobin, those molecules, even though they don't have a big diffusivity, they can kind of shake around a bit and they can transfer oxygen to nearby hemoglobin molecules. And so effectively, you get diffusion of oxygen bound to myoglobin. And S is the saturation of myoglobin with oxygen. Okay. So again, you have a time-dependent equation, and if you want to, you can solve it as a steady state. This now, here's this mass transfer coefficient K. So if you have a difference between blood PO2 and tissue PO2 near the wall, then you get a wall flux J that you can put into your vessel convective transport equation. This same wall flux also has to be the boundary condition on the tissue, right? So where the tissue meets the vessel, there's a flux, let's say, of oxygen into the vessel into the tissue from the vessel. And so the gradient in the tissue times d alpha has to equal that flux. Okay, so this is your boundary condition here. Okay, and so these equations are basically given in or have been put into finite difference form and solved in that way. So there's many ways to do that. Sanjay will talk about that. But you can use a time-dependent or a steady-state method if you want. What we've mostly done is use a time-dependent method. And if we want steady-state results, we just solve for a while and we get steady-state. Okay. So what I'll talk about is only steady-state here. All right, maybe I'll stop. Oh, I see some chatting. Any questions about this model here, these equations? No? Okay. Let me go to the application and then maybe you'll have questions. Okay, so I'm talking about applying that transport model to basically bunches of capillaries in 2 disease states. One is sepsis and one is prediabetes. And so in this case, it's sepsis, and we use the model to study an array of capillaries. So we also see these lines, oxygen is coming from this side, it's flowing in, it's diffusing into the tissue and being consumed. And there's some heterogeneity in where these vessels are. Okay, so they're just parallel capillaries, but they're spaced irregularly. And so up here now, this is the control case where you have a lot of capillaries, healthy tissue and a normal consumption rate. And you can see that, so red is high O2. You basically have pretty high O2 everywhere in that domain. But now as you go to sepsis, so this is kind of average sepsis, you start to go down here in oxygen and you get certain regions that have lower PO2. And then finally, this is an extreme sepsis case where you've lost, I think, 40 or 50% of your capillaries, and you actually have an increase in O2 consumption, which is a little strange. But the inputs to this model come from experimental data. And so then you get some regions now with very low O2. And so the thought is you may start to get some hypoxia locally when you have sepsis, when you have severe sepsis, okay? So that was from 2004. And then pre-diabetes, this is from 2010, we did a similar model. And again, we put in experimental data for losses of capillaries, changes in blood flow in those capillaries. And these two pictures are sort of cross sections through tissue blocks showing what the PO2 looks like for a control case and a pre-diabetes case. And you can see that there's heterogeneity in capillary spacing and all that, and in oxygenation for the control case. But in the pre-diabetes case, this heterogeneity results in some lower oxygen regions. Okay? And so this is kind of what you expect when you lose capillaries, and maybe you have some changes in flow. And so that's what this model told us. And so now, if we take these O2 distributions and look at them as histograms, basically, then this red one is control PO2, and it's very sort of sharply focused around, that is maybe 48 millimeters of mercury, whereas pre-diabetes from here, you get more of a spread in PO2, and you get a mean that's lower. It's maybe 39 or something. So you've dropped in mean PO2, and you've gotten more of a spread, and you're getting to lower PO2 values here. The two dashed lines are cases where we've sort of tried to fix the defect in pre-diabetes, and so we fixed it just by increasing total flow. And so I think the green is if we double flow, and the orange is if we go, sorry, I can't read it here. But basically, if you increase flow enough, you get close to the normal PO2 distribution in the tissue. Okay? And so that's what we found from this model. All right, so let me take any, before I do that, let me take any questions on this, if anyone.
I believe Yusuf has a question.
Okay, Yusuf?
Yeah, so I'm more interested in, I'm young as well, so I don't know much, but I'm interested in why diabetics have this issue. Like, because I know some diabetics, they wear socks, right? Like they have special socks. Is that correlated?
Well,
Right.
so, okay, so this study was pre-diabetes. And so even though you may say, if you look at this, the numbers aren't very great, right? From 48 to 39, that's not a big change in PO2. But this is in before anyone would be diagnosed with diabetes. If we did this in a diabetic case, I think this capillary density would be much lower, and this would be much more to the left. You'd have much lower PO2 values. And so in diabetes, once you have high glucose in your blood, and you've lost lots of capillaries, then you have an even worse time delivering oxygen to tissues. And if you actually have a very low capillary density, then if you try to exercise or something, you can't get oxygen far enough into the muscle. And so that's why you get like leg cramps in diabetes and things like that. And so again, things would be more severe in actual diabetes. But it already starts. So pre-diabetes is you have high glucose, but normal you have high insulin, but normal blood glucose. So it's like your body's making up for a problem, but eventually you get full-blown diabetes. So I don't know if I answered your question, but what do you think?
Prof. Keith has a question.
Okay, Keith?
Again, just a quick one, Dr. Goldman. I never think about it often because I don't see the effect of myoglobin in equations all that much, but on the previous slide, the equations you displayed, I know the P50 of myoglobin is quite low. I think it's around 5 millimeters mercury. So we never end up going that low, but does it present a big difficulty implementing it computationally? Like, does it add more of a computational burden?
Let me see if I can go back here.
Because it would be quite nonlinear, I think, because it has the
Yeah, well,
dependency on that
no,
form.
so in a finite difference code, it doesn't cause too many problems because, you know, you can handle the nonlinear part. I mean, especially a simpler code, like if you're using an explicit method, then it's not as bad. If you're using an implicit method, it would, I think, cause you to do more iterating. And so it could be a little bit of a problem. But I agree, Keith, that if you know your PO2 is staying above 10 or something, then you probably could drop the myoglobin and nothing would change. And especially, and also if you want steady state, I think this term, you can see, right? If you're at steady state, this term doesn't do anything. So you could get rid of it. Even though you keep this one, it still, I think, would simplify things a bit.
Cool.
Okay,
Thank you very much.
sure. Go back to here. Any other questions on this? This is just a demonstration of these oxygen transport models. And that they can give us tissue PO2 distributions, which are hard to measure, you know, at this scale. And it's easier to measure what's going on in capillaries, but this lets us estimate what's happening in the tissue. Okay. So the next thing I just was going to do is two quick in progress topics that may be interesting to people. And so one has to do with, you know, if we measure these arteriolar networks and even the venular ones, we've got these giant spaces. And we can see capillaries there, we can see muscle fibers. So we know something's there, but you know what we've measured doesn't cover that. And so there could be a reason why we don't see vessels there, like they dive into the tissue and we can't see them, or they're very small, or maybe during this experiment, those arterioles were constricted for some reason and were hard to see. So there's a reason why we might want to sort of complete this network. And then there's also a reason for theoretical studies where we might want to construct networks like this, you know, from zero. And so we've started to do that. And so the green here is, I think it's this red network on the left. It's just flipped upside down. And so then the blue is vessels we've added. Okay. And so for this work, it was a very simple algorithm where we just looked at tissue points that were far from a vessel, and then we started growing vessels, you know, just constructing vessels in that direction. And so by doing this for a few iterations, we've increased the vessel density in certain places where vessels were missing. And then we can go and we can estimate diameters of those vessels that we added, and then we can try to do a flow simulation. And so that should allow us, now, for example, you know, Dr. Frisbee asked about tracer washout kinetics. So if we just have this network, or if we just have, if we just have the green network here, maybe that won't give us the right washout kinetics because we're missing a lot of things. But maybe if we add these blue vessels, we'll get a better washout kinetics, and we'll maybe get better oxygen transport and things like that we should have for this this network. So that's one of the things that are in progress. There's different approaches that we're going to try, but that's where we are on that. And maybe I'll just do the next thing. So the other thing that's in progress is developing a continuum model. So everything I've shown so far for oxygen transport was using discrete vessels. But there are so many that we may want to try to do a continuum description. So, let me go back to here. So even though I've put in arterioles, there's still lots of gaps. And in those gaps, there's going to be capillary beds, right? So there will be lots and lots of capillaries, and probably too many to measure them all. I mean, some people try, but if we want to do something at this scale of this whole network, we may want to approximate the capillaries by a continuum rather than putting in discrete vessels. And so to do something like that, we have to develop a method. And so with Keith Appis and then some contributions from Rashi Vijay, we developed some equations that we can use to describe continuum transport. So if we have capillaries and we assume they're spread out in a sense, and we just have two two compartments. One is tissue with a PO2, one is blood vessels or capillaries with a saturation. Then we can have transport equations, and this is steady state. So this is like a diffusion term. This is a convective transport term. These are the two transfer terms, like mass transfer coefficients, the Ks. And then what we can do is can try to validate this by comparing it to a discrete model. And so that's what we did here. Maybe hard to see, but this is tissue PO2 in this kind of 2D domain. And the dashed lines are, right, so the dashed lines are from the continuum model, and the solid lines are from a discrete vessel model. And so for the tissue and for the capillaries, we compared the results for discrete and continuum, and you can see that they're pretty close now. And so if the continuum model does this well, then it's a good sign that we can use it to actually study oxygen transport and maybe flow regulation in large networks. And so that's the objective eventually. Okay, so that was all I had planned, and I'm willing to take questions on anything. Thank you.
That was very informative, Dr. Goldman. Thank you very much. So people, please do take advantage of this time and ask any questions, initiate discussion. Yeah. So make full use of the time provided to you. You can unmute yourselves and ask the question. I'll start. I'll start the discussion. So Prof, I wish Dr. Frisbee and Dr. Boss Frisbee were here, but I'll go on treating them as our users of the end product, the software platform, whatever it is that you will call it. Would you be kind enough to make a suggestion on how to include one most relevant JSIM model from the University of Washington? So what is the most relevant model that the University of Washington are providing, which will be useful in our work over here?
Well, probably, I mean, they have an oxygen transport model, I think, that it could be used. That's a possibility. I actually have to look at more of their models, though. I would say one thing I could say is maybe you and I and a few of the students need to sort of comb through their models, because I don't really know everything they have.
So I came to the same conclusion. We should have a designated person who goes through the models, who has the list of papers or your work from recent past or even distant past. And we can then put that together and see how we can uptake the JSIM models into our work fruitfully. Yeah. Perhaps someone will also bother to make a list of Dr. Frisbee's papers. So that was a question, and I agree that we should have a designated person with assigned time to start the process. Could you explain to all of us the motivation for why you developed the continuum model, even as the discrete model already exists?
Good question. Well, I'll just go back here for a bit. So basically, in a network of this size, right? So this is 2 by 2.5 centimeters. So I can count all these arterioles. It's not fun, but I can count them all. And I can easily get my flow simulation model to do a flow simulation. But to do oxygen transport, there's basically, I estimated the number once, it's something like 100 million capillaries or something. And so if you wanted to put all those capillaries in, you would have to arrange them. You would have to figure out what the flow is to each capillary. So there would be a lot of work. You'd have to make a lot of assumptions. I think you couldn't probably measure them all. And so to do proper oxygen transport to tissue, you would have to know, where each capillary is, how much oxygen it's getting, and then do the transport to the surrounding tissue. Maybe you'd find some interesting things, but it's a lot of work, and I don't know how feasible it is to really do that in detail. But a continuum model, you could just say, this vessel supplies some region of tissue, and then you could say, okay, that flow gets distributed. I can discretize that volume any way I want. It's convenient, like for finding differences, and I can compute the oxygen distribution that comes from the supply here. And so then I think you could basically distribute over all the terminal vessels that you eventually place here. Each terminal vessel, you can just calculate how much oxygen is delivering to a certain region and get tissue PO2 distributions.
And so if a certain specific small region of the vasculature is of interest, we can always go from the efficient continuum description to the discrete description.
That's right. That's right. So if we think there's a problem in one place or any place that we want, we can pop in a discrete model, and the rest of it we can model with a continuum if we like.
Yes.
Yeah, that's good. Yeah.
So Lawrence has a question. Lawrence, go ahead, unmute yourself, speak up.
Hi, Dr. Goldman. I'm just curious, if there was like a plaque in like an arteriole, would you still consider pumping more blood flow, or would you just remove the plaque? Like, which one is more efficient? Like a more efficient solution?
A plaque, like in a small vessel here, let's say?
Yeah, like it's restricting like the blood flow.
So it's actually like an occlusion of this?
Yeah.
So if there's an occlusion, probably, there's some alternate flow path to get you most of the same flow somewhere. And that would eventually, there probably would be some angiogenesis. Eventually you would develop, so maybe, like say these two vessels would eventually connect and become larger, and you would have an alternate way to deliver flow to the same area.
Oh, okay. Yeah, I see.
I think that's probably what happens, but depending on how large the vessel is, it could take time. Like you could have a lot of damage downstream to tissue, and then you would have to have a regrowth process.
Yep, I see. Thank you.
Yeah. But Lawrence, just let me follow up a little bit. I mean, if you had a small vessel here that was occluded, in that case, maybe more flow to the surrounding ones would make up for things. And even capillary networks, they're not really disjoint. If they're nearby, there's usually some interconnections, so they could share blood flow potentially. Clara?
Yeah,
Yes, Clara, go ahead.
thank you. I just wanted to sort of build on Lawrence's question as well, but more so focused on just I guess, because I'm interested in kidney disease, and I was wondering, I know you mentioned you do work on muscle mostly, but would, I guess, tissue during the hemodialysis, if and in relation to, I guess, ischemia that may or may not be happening, what kind of changes are happening on the microvasculature level?
Sorry, do you mean in any tissue, or do you mean in the kidney, or?
When
I
there's
guess
a dialysis unit attached.
Yeah,
What
in
are
any.
the-
Mhm.
What would be happening in ischemic tissues?
Yes.
So yeah, so I guess, I mean, blood flow, like let's just say blood flow decreased to this network, then there could be certain regions that are sort of in the worst shape. I don't know where they are ahead of time, but there could be, let's say over here, is actually suffering the most. And so it would try to signal for more blood flow, and maybe some cells would be damaged or die from hypoxia. And so then if they didn't die, then I guess there may be some angiogenesis, increasing the flow capacity in that area. So I think it's kind of like either there's enough localized hypoxia to do damage. If somehow the area survives, it may try to adapt, I think, to try to get more flow in the future. But in a lot of things like diabetes, sepsis, probably in dialysis injury, it's very heterogeneous, right? So it depends on how this network reacts when you cut its flow below what it really needs, and where where the damage happens, which it may be hard to find the damage.